Hello, my name is Rocio Zaldivar, and I am a McNair Scholar at the University of Florida. My research mentor is Dr. Arya Shragi. Together, we're conducting research to characterize the Francisella type 6 secretion system apparatus. Francisella is a bacterial pathogen that causes a disease named tularemia that leads to a rapid time of death. This bacterium has a circular chromosome where a cluster of genes called the Francisella pathogenicity island is required for virulence. The cluster of genes encodes a secretion system used to export these toxins from the bacterium and into the host cell. We don't know the nature of this secretion system. We also don't truly know the identity of the components of the apparatus, but we know some of the proteins encoded by the Francisella pathogenicity island share homology with other bacterial type 6 secretion systems. The best characterized type 6 secretion systems are those from other bacteria. This image to the right is the canonical type 6 secretion system apparatus. Those systems are very well characterized. They are complex systems composed of at least 17 proteins to make up the apparatus. The components that make up the Francisella type 6 secretion system apparatus lack homology with the apparatuses from other bacteria. Based on that, we know that Francisella apparatus is unique, but beyond that, we just don't know a lot about it. The first image is a type 6 secretion system apparatus, and our goal is to characterize the protein-protein interactions amongst the components of the type 6 secretion system. By characterizing the protein-protein interactions, we will begin to understand how the apparatus functions. Defining these mechanisms will lead to therapeutic avenues in the future. In the short time that I have been in the lab, we have already made significant progress towards this goal. We have constructed plasmids that append a VSVG epitope to the open reading frames of several genes encoded in the Francisella pathogenicity island. After construction of the Francisella mutants, we verified the correct placement of the epitope in the genomic DNA by using PCR. We use primers that anneal at the epitope tag and downstream of the gene and ran the PCR product in an agarose gel. If you look at this gel, you only see a PCR product with the mutant, not with the wild type. This confirms the placement of the tag at the 3' end of this gene. Moving forward, our goal is to use this strain to identify components of the type 6 secretion system that interacts with this epitope tagged protein. To do that, we will sediment antibody conjugated beads from the lysate to precipitate tagged proteins and determine the identities of interacting proteins. This process will reveal the protein-protein interactions of the Francisella type 6 secretion system, and we'll begin to characterize how the apparatus works. Our future plans are to tag the remaining genes with the VSVG epitope and characterize the protein-protein interactions in the apparatus. The Francisella pathogenicity island is a cluster of genes found in the circular bacterium chromosome. We have successfully tagged 11 genes out of the 18 found in the Francisella pathogenicity island. The tagged genes are denoted with the red color. I would like to thank Dr. Arya Shragi for always being actively ready to help and to answer all of my questions, and for dedicating so much time in this research project. I would also like to thank Danielle Hovey and Adele Rodriguez for their dedication and hard work for this project. I would like to thank them all because without them, this project would not be moving along as efficiently as it is. I would also like to thank the McNair Scholars Program.